Hello, myself Dr. Rashmi. I am coming from Sri Nilakanta Government Sanskrit College, Pattambi, which is affiliated to University of Calicut, Kerala, India. I am here to present our findings on growth, structural adaptation, and physiological fluctuations of Alternanthera tenella Colla towards cadmium stress. This is a recent report by Hugh et al. In 2025, which shows the global distribution pattern of cadmium contamination. We can see here a metal-enriched corridor which starts from southern Europe into the Middle East to southern Asia. This is an area where high agricultural productivity overlaps with mining as well as industrial activity with very limited regulations. So the result is that between 1.9 and 1.4 billion people lives in this region is severely affected by various health conditions as well as at ecological risk. So this study and also this red regions also shows the cadmium concentration in agricultural soils which with the increase above the threshold level. So these studies insights or justify the urgent need of remediation of this contaminated soil and a suitable candidate species, which is a plant or a microbial species, may be needed to mitigate this heavy metal stress from agricultural soils. Among the various heavy metal stress such as cadmium, chromium, lead, arsenic, cadmium is very, very dangerous. It is considered as a group 1 human carcinogen by IARC, group A carcinogen by WHO, EPA, EC. It mainly released as industrial effluents into the rivers of human consumption by mining, smelting, e-waste or synthetic phosphate fertilizers. The main threat is that it exists in soil as chromium 2 plus or cadmium 2 plus or cadmium chelate, but it is highly bioavailable to plants for root uptake and translocation. In turn, it reaches into the diet of human as well as other biological organisms. Humans also, the cadmium contamination is very threatening, life threatening. It affects brain, heart, lungs, liver, kidney, etc. etc. So the famous Ita disease is also caused due to cadmium contamination. So why Alternanthera tenella? Our preliminary studies in Valaya River bank along the industrial affluent areas of Kanchikode, which is the second largest industrial hub area in India. You can see that how heavily contaminated the Valaya River near the Kanchikode. It is almost the water turns blackish in color. We have analyzed the cadmium concentration in the soil samples and it is very dangerous level 2505 ppm. So this is Alternanthera tenella which flourish wells in the contaminated sites along the Valaya river bank and it is the most dominant species on our phytosociological analysis. So we also analyzed the cadmium concentration in root and shoot and there is a much higher amount of cadmium reaches the shoot of shoot of Alternanthera tenella. So we want to know the molecular mechanism behind the plant stress adaptation to cadmium contamination because it is a very underexplored plant species for phytoremediation. So understanding the molecular as well as the cellular adaptation of this plant species against the cadmium stress is very important to develop it as an ideal species for phytoremediation. So our objective is to explore the efficiency of Alternanthera tenella in cadmium phytoremediation, shedding light on its morphoanatomical and physiological response to cadmium stress. So we have set up our experimental conditions. We have collected the Alternanthera tenella, healthy plants of Alternanthera tenella from uncontaminated sites and grown in Hoagland's medium, and equally grown plants of same age as well as same number of nodes as well as same number of leaves has been selected and set up as 5 replicates. And for the experimental conditions, we have selected the cadmium concentration from 0 to 200 micromolar. And 170 micromolar has been standardized as the effective concentration of stress to Alternanthera tenella because after 170 micromolar concentration, the plant show heavy decline in growth as well as necrosis and death. And we monitor the plant in cadmium given Hoagland's medium. The cadmium is given as salt, cadmium nitrate in cadmium 2 plus form. And we monitored the growth pattern of plants in 0, 7, 14 and 21 days intervals. And root length, shoot length, dry weight, fresh weight, leaf area has been monitored as morphological parameters. And significant reduction in various growth indices were noticed. The root length has been decreased by 41%, and shoot length also there is a gradual decrease, a severe decrease in shoot length by 22% in 21 days of stress condition. These growth inhibitions may be mainly linked to disrupt mitosis, reduced mineral uptake, or impaired photosynthesis. We have to study it further to validate our hypothesis. The cadmium stressed leaf area also shows a severe decline by 48% at 21 days. And an overall declining tolerance index indicates a progressive stress in plants in 21 days of stress conditions. So this growth impedes with any ultrastructural modifications we want to know. So we look into the ultrastructure details of root, stem and leaf by SEM analysis. This is a SEM analysis of root under control as well as cadmium stress. And we can see that the root tissues shows a reduced xylem diameters with a distorted parenchyma and thickened xylem walls, and electron dense depositions were observed in cortical as well as vascular region. And in stem also, the cortical cells exhibited distortions and occlusions with the xylem wall thickening and with the xylem wall thickening and vessel diameter reduction to minus 32 degree, which reflect inhibited vascular transport and severe growth inhibitions and mineral uptake. This is the enlarged view of cortical cells of cadmium treated Alternanthera tenella stem. As I already said, the xylem wall has been shown to be thickened in a stem with an overall decrease in xylem vessel diameter. When we compare the leaf and stomata, in leaf also the trachary occlusions has been seen, whereas the stomatal depositions were also noted with an overall xylem wall thickness increased to 4.11 micrometer, whereas there is an inverse proportional of vessel diameter with a minus 23 degrees percentage. Stomatal behavior, we can see that there is a fully open widened stomata in control, whereas in treated plants, majority of stomata seems to be partially closed or completely closed with thick guard cells and deposits all over the leaf area. So when we compare the xylem diameter and thickness of xylem wall in different tissues of control and cadmium treated Alternanthera tenella, in root you can see that the diameter of xylem vessels increased to 9 and in stem 28, decreased to 28, and in leaf also there is a considerable decrease in the diameter of xylem. But the thickness of xylem vessels is found to be increased in all root, stem and leaf. In root it is 3.22, stem it is 7.82 and in leaf it is 4.11. So to conclude, we have found that the cell wall thickening and lignification in xylem vessels restrict cadmium translocation, act as a physical barrier and reduced radial water flow, which is in line with the report by Pandey et al. In 2022. But before concluding, we want to support it by FTR analysis, whether the cadmium interact with the cell wall components such as cellulose, lignin and pectin result in which act as a physical barrier. Before FTIR, we go for IDAX analysis of Alternanthera tenella root. The IDAX, there are three regions. We have divided our whole root and stem leaf into three regions, region 1, 2 and 3. Region 1 consisting of epidermis and cortex, region 2 endodermis and vascular region, and region 3 as pith. When we analyze the essential as well as essential macro and micronutrients, we found that a majority of macronutrients were found to be reduced in treated plants where compared to carbon and oxygen is found to be reduced in treated plants when compared to control, which shows an increase in sugar translocation towards leaf and stem. And when comparing the essential nutrients, there is no noticeable amount of aluminum or silicon in any of the treated plants, whereas the presence of copper has been found in endodermis and vascular regions of cadmium treated plants. In Alternanthera tenella stem, IDAX shows that the presence of potassium in epidermis and cortical regions of cadmium treated stem, and also sulfur and calcium also present in all treated in all the three portions of cadmium treated stem. And there is presence of copper is restricted only to the outer region, and there is no presence of zinc in cadmium treated plants. And in leaf also, there is no phosphorus in outer portion of cadmium treated plants, whether it is restricted to the endodermis and vascular region, but the presence of sulfur is noted in cadmium treated plants. And the aluminium also present only in the control treated pith region, and the presence of silicon is noticed only in the cadmium treated endodermis and vascular region, where there is no presence of zinc, chlorine or iron in any of the treated region. So the decline in carbon shows an increased sugar transport, and decrease in potassium shows the partial closure of stomata. And the enhanced level of phosphorus may be due to enhanced ATP synthesis result in ROS production. And cadmium can be replaced by magnesium in chlorophyll, thereby decreasing the photosynthesis. So we move forward towards the FTR analysis of root, stem and leaf. There is a significant peak shift in 3000 to 3500 region, which is majority of OH and NH region, 1640 to 1600 region, which is of keto region, C double bond O, and 1000 to 1500 region also, which indicates the cadmium binds with the hydroxyl, amino, carboxyl and phosphate group of cellulose, lignin, pectin, etc. Maybe, and this binding alterations suggest cadmium interaction with protein, lignin, pectin and cellulose, thereby impeding the transport of cadmium to upper portions of stem and leaf. It acts as a structural barrier also. So whether this effect in photosynthesis, whether this impaired translocation of nutrients have any effect in photosynthesis, we look towards the photosynthetic pigment production by mainly chlorophyll and carotenoid content. There is an overall reduction in chlorophyll and carotene content. There is 39% of chlorophyll over total chlorophyll reduction after 21 days, and also carotenoid also shows a significant decline in production after 21 days. Then we analyze the total protein and soluble sugar under controlled and cadmium stress conditions. Soluble sugars initially shows a upregulation at 7 days, but then it declines significantly after towards 21 days in cadmium treated tissues. This is mainly due to the initial increase in sugar production, followed by the impaired photosynthesis and carbohydrate translocations from source to sink, such as root. So the total protein also shows an earlier rise due to the synthesis of early stress proteins such as heat shock proteins, maybe due to the production of heat shock protein, phytochelatins and antioxidants. But gradually they show a decline, maybe due to the protein degradation and reduced biosynthesis. The proline and MTA were also analyzed. These two are the major indicators of stress in plants. Proline is an osmoregulator which is found to be increased in almost all stress conditions in plants. Here also there is a significant increase up to 7x in roots by day 21. It acts as a osmoprotectant and metal chelator. Early accumulation in leaves and stem also suggests a rapid defense response of Alternanthera tenella towards cadmium stress. Malondialdehyde or MDA, it is a result of indicator of lipid peroxidation and oxidative membrane damage. We also found an elevated level across all plant parts under cadmium stress, which reflects an increased ROX production due to cadmium toxicity. Total phenol and flavonoids were also analyzed. There is a gradual increase of flavonoids and phenols in all plant parts, which shows a stress response to Alternanthera tenella by producing the secondary metabolites under cadmium stress. Finally, we have analyzed the cadmium concentration and accumulation in various plant parts of control and cadmium treated plants on 21 day. We found that highest cadmium concentration were noted in roots 317.25 milligram per kilogram dry weight, followed by stem and leaf with a BCF bioconcentration factor of 8.39, biological accumulation coefficient of 16.6 and translocation factor of 0.5. This high root retention and TF value lower than 1 suggests Alternanthera tenella as a suitable candidate for phytostabilization rather than phytoextraction. So to conclude, these are our results. This is Alternanthera plant. When we fed with cadmium divalent ion, it absorbed the cadmium divalent ion from the medium and it accumulates major cadmium in the root, and meager amount is transported through the root and to the stem and leaf because Several structural barriers exhibited by plants, by xylem wall thickening, growth retardation, cell structure distortion, closing of stomata, depositions in root and leaf shells, etc., which all result in decreased nutrient uptake, increase in stress metabolites such as proline, MDA, reduced pigment production such as chlorophyll and carotenoids, and sugars also. There is a severe shift in FTIR such as the transition metal interaction of cadmium uh, with amine groups as well as keto groups of maybe proteins, pectins as well as cellulose in the cell wall. So this work was supported by DST Serb Sure, India, under the project title Understanding the Mechanism of Heavy Metal Detoxification in Alternanthera tenella Colla to elucidate its phytoremediation potential. I would like to acknowledge Sir for funding our project, uh, our college principal and VC University of Calicut for providing all necessary facilities, and the most important, our team members, the plant physiology and molecular biology lab members, for their support. Thank you. I will be happy to answer any questions or queries. Thank you.